Bismillah Rahman Rahim. Hello and welcome back, friends. Welcome to another video lecture by Kami Piaruchi. In this video lecture, we are going to talk about antigen presenting cell or monocytes, or we can say macrophages and dendritic cell. So let me write antigen presenting cells, or we can say macrophages and dendritic cells. Dendritic cells which are related to monocytes. Now in the previous video lecture, if you guys remember, we talked about the overview of immunology, or we talked innate and adaptive immunity, and we also talked the hematopoiesis. So this is another video which is also regarded to what? Which is also regarded to the immunology, and we are going to talk about antigen presenting cell, means macrophages, dendritic cell. Macrophages and dendritic cell, actually it is related to the monocytes. Monocytes are actually converted into what? It is converted into a dendritic cell and macrophages. We call macrophages and dendritic cell antigen presenting cell. Why? Because they can present the antigen. They can break down the pathogen into small fragments and then show it to the to other amino cells. Okay, now B cell is also antigen presenting cell, but why I not write here B cell? Because I will explain the B cell in some other video lecture, not here, because here the monocytes, actually I am explaining the monocyte, and monocyte are usually related to this macrophages and dendritic cell. That's why I didn't write the B cell, although the B cell is also antigen presenting cell. Okay, now inshallah I will explain the B cell in some other video lecture. Now to explain this dendritic and macrophage, first of all we need to understand the monocytes. Let's first of all explain the monocytes. So let me draw here the monocytes. Monocytes, if we talked about the monocytes, so monocytes are usually, it is usually oval in shape. So it is not usually circle, usually oval in shape that I draw. It have nucleus which is bean-like shape, or we can say a shoe-like shape, or we can say horseshoe-like shape, bean-like shape, or kidney-like shape. So let me draw. Now this is what? This is the nucleus. This is the cytoplasm area. This nucleus, as you are watching, it is look like a bean-like shape, kidney-like shape, or we can say it is look like horse-like shape. Okay, this cytoplasm, monocyte cytoplasm, are usually clear cytoplasm. Whenever you stain it with the GIMSA, okay, we usually stain it with what? With GIMSA. Try to remove the spelling mistake by yourself. Now GIMSA is a kind of stain, is a kind of color which we usually provide to this monocytes. Due to this way, this nucleus will take this color, means this GIMSA stain, and due to this way we can easily visualize it with the help of light microscope. So we can easily see it with the help of a light microscope. They have cytoplasm, but clear cytoplasm. Okay, but in some books they says that they have cytoplasm which contain granules. Usually when you see this monocyte, so monocyte don't have granule. Okay, this cells is also called agranulocyte because they don't have granules. Okay, usually they don't have granules, that's why we call it agranulocytes. Their nucleus can occupy the whole cell. Okay, and it is, this cell is the largest cell. What? This is the largest cell, or we can say this is the largest leukocytes. Leukocytes. When you see it under the microscope, and some other cell, when you also see the other cell under the microscope, so you can find this cell in a large number, or you can find the size of this cell in a large. Okay, that's why we can say that this is the largest cell among the leukocytes. Moreover, this monocyte, if we talked about the monocytes, so their diameter, as it is the largest cell of the leukocyte, so their diameter will be, their diameter will be 14 to 18 micrometer. Usually the monocytes are developed and synthesized inside the bone marrow. So they are usually developed inside the bone marrow, mature or synthesized inside the bone marrow. Once they are synthesized from the bone marrow, then they release out from the bone marrow, and they will circulate inside the blood almost 8 hours. Now whenever they are circulate inside the blood, then we call that it is monocyte. Okay, 8 hours inside the blood. After 8 hours, they reach to the specific site of the body. Sometimes they reach to the skin area, mucous area of the lungs, nose. At that specific area, these monocytes will be reached. But before that, they are circulating 8 hours in our blood. Okay, their lifespan, if we talked about their lifespan, so the lifespan of the monocyte is 4 to 5 days. Now moreover, these monocytes sometimes they have surface receptors, as I am drawing. So they have also the surface receptors, which are usually involved to recognize the pathogen. Okay, so this is the surface molecule, or these are the surface receptor. Surface receptor. Okay, so that's a little bit about the monocytes. Now if we talked about this macrophages, dendritic cell, as well as if we talked about the monocyte, so all these 3 cells, although this is not the 3 cells, actually these are the 2 cells. This one monocytes is actually divided into 2 more cells, which we call it dendritic and macrophages. If we talked about their properties, so both these 3 cells are what? These 3 cells are actually phagocyte in nature. They are what? They are the phagocytic in nature. So these cells are usually phagocytic in nature. And one thing more about this cell, these macrophages and dendritic cell, these 2 cells are what? These 2 cells are usually what? These 2 cells are usually antigen presenting cell. And one thing more, they have what? They have a receptor which we usually call that MHC. MHC means MHC1 and 2. Now let's a little bit talk about this MHC1 and MHC2. Don't worry about the MHC1 and MHC2. I will explain it in much more detail in some other video lecture, but not here. But here I will just a little bit clear your concept about the MHC molecule. MHC molecule are a kind of receptors that are usually present in all nucleated cell. Okay, so we have dendritic cell, and we have all amino cells. So all amino cells have what? This MHC1 molecule. If we talked about the macrophages and dendritic cell, they have MHC1 molecule. Except MHC1 molecule, they have also a unique molecule which we call that MHC2 molecule as well. So they have also what? They have also MHC2 molecule as well. Now what are the function of these 2 macrophages and dendritic cell? So let's say this is a cell. Means let's suppose this is macrophages, or let's suppose this is what? This is a dendritic cell. Let's suppose maybe macrophages or dendritic cell. What actually happen? They, as I told you, they are involved in phagocytosis process. So they will engulf, this is antigen. What? They engulf the antigen. Now this antigen will internalized. After that, a phagosomes are formed. Okay, so this is what? This is the phagosomes are formed. Now this is our antigen. This is what? This is our, this phagosomes will be fuse with the lysosomes. So let's suppose this red one representing the lysosomes, and this is what? This is our vacuoles, which we call that phagosomes. Vacuoles or phagosomes. These vacuoles have what? These vacuoles have this antigen or pathogen. Now this pathogen will be degraded, as you are watching. Why? Because this lysosomes have a kinds of enzyme or a kinds of acid or molecules which we call that lysozymes, hydrogen peroxide, or degraded enzyme, or hydrolytic enzyme as well. Due to this way, they will cause the degradations of pathogen. So then the pathogen will be degraded. Now moreover, after this pathogen is degraded, then they will transfer this pathogen to their surface. So after that, this pathogen will be transferred to their surface, and we call that what? Now these are, let's suppose, the fragments. These pathogens are converted into small fragment, into small pieces, and then they will be transferred to this surface of this antigen presenting cell. And this area, as you are watching, we call this what? We call this MHC2 molecule. So they have what? They have also MHC1 molecule as well. This is, let's suppose, MHC1 molecule, and they have also this MHC2 molecule as well. So this is how the phagocytosis process of the antigen presenting cell. All antigen presenting cell perform just like that process. Means B cell, dendritic cell, and macrophages perform like that processes. They will present their they present this kinds of fragments, pathogen fragments, to the other cells. The other cells will comes and recognize it. Means I'm talking about the T cell. Once they are recognized, so then the other macrophages and more dendritic cells are activated and reach to this specific area, and the problems or the disease or some other problems are under the control. Okay, so that's the main point, that's the logics, that's the concept. Okay, let's explain this macrophages in a detail. Okay, so that's all about what? That's all about the job of antigen presenting cell. So let's explain the macrophages. So usually, if we talked about the macrophages, so the macrophages structure will be look like in this form. Now, this monocyte is converted into dendritic cell, or means this monocyte will be converted into macrophages or dendritic cell on that time when there is infection chances, when there is inflammation chances. Then macrophages will be formed from this monocyte. Monocyte is converted to macrophages. Again, the macrophages are what? It is the largest cell. What? It is the largest cell. So let me write, this is what? This is the macrophages. Now, these macrophages have also the phagocytosis property. And if we talked about the macrophages, so they have what? They have also surface receptors. So they have surface receptors, two kinds, although they have many kinds of the surface receptor, but here I will draw the three basic surface receptors. So the first, they have the receptor which we usually call that what? MHC1. As I told you, all nucleated cells have the MHC1, but except that they have also what? They have also MHC2. So let me draw here. So this is what? This is the MHC2. MHC2. Now this MHC2 are not always present. This MHC2 will be synthesized when the pathogen are converted into small fragment. Then this MHC2 will be synthesized, because here the pathogens are presented on this MHC2 molecules. These macrophages have some other receptor which we call as scavenger receptors. So they have another receptors which we call that scavenger receptors. So this is what? This is the scavenger receptors, and we usually call that CD14. Now this is cluster destination. CD means cluster destination. So they have another receptor which act as a scavengers. Okay, that's why we call that a scavenger receptor or CD14. What does scavenger means? It means that they can recognize the death form of the pathogen, or they can showcase the death form of the pathogen. They usually recognize the death form of the pathogen. Okay, whenever, let's suppose, this cell is presenting an antigen, and this antigen is in a death form, so this CD4 will recognize it. So that's why we call that scavenger or scavenger receptors. They have also more organelles, and moreover, they can cover the plasma membrane, as they are what? These are the plasma membrane. They have also what? They have also the plasma membrane. So they have also what? They have also the plasma membrane, and this plasma membrane is covered by the microvilli. Now this portion, this one, as you are watching, this portion are called what? This portion are called microvilli. So let me write microvilli. Now, the plasma membrane is covered by the microvilli, and these cells have more organelles than the other amino cells. That's why we say that it is the largest cells. Okay. Moreover, they have another surface receptor which we usually call it CR3 receptor. So let me draw the CR3 receptor with this green, black, blue, with this blue marker. So they have also what? They have also CDR3 receptors. So this is what? This is CR. Sorry, they have also CR receptors, CR3 receptors. Now, CR means complement receptors. Okay, complement receptors 3. They have what? They have complement receptor 3. These receptors, usually recognize the cell, or we can say the pathogen component. Usually, which kind of the pathogen component they are recognizing? They are usually, recognizing the lipopolysaccharide. So let me write. They usually recognize the lipopolysaccharide. We can say that they also recognize a fungal cell wall, which we call that zymosin. So they also recognize the cell wall of the fungi, which we call that zymosin. Moreover, they recognize viral nucleic acid, and foreign DNA. So these things are recognized. These are what? These are the microorganisms, or we can say these are the pathogenic component. These are what? These are the pathogenic component. Pathogenic components, which is actually sometimes recognized by CD14, CR3, MHC2, not MHC1. Means these kinds of things, these kinds of receptors recognize these things. Once it recognizes this kinds of component, or let's suppose, whenever they have CD14 and CR3, then both recognize such kinds of molecules or such kinds of things which are present on the surface or inside the cell. Then what actually happen? They will, coat it the cell. So let's suppose if this is the pathogen. So this things is also, come closer. This one CR3 is also come closer. This MHC1 is also come closer. These three receptors are come closer to this antigen, or we can say pathogen, and they will enhance the chances of phagocytosis. As these three things are present, so with the help of these three receptors, they they can increase the enhance, or we can say they increase the chances of phagocytosis, and we call it opsonization. Remember, opsonization is that phenomena in which whenever the pathogen are coated with the antibody, and if the antibody and it is phagocyted by it is engulfed by the phagocyte cell, we call that opsonization. So then opsonization phenomena occur. Okay. This macrophages is present in different sites of the body. They are present in the brain, they are present in the liver, they are present in the bones. Whenever the macrophages are present at that specific area, then the macrophages has different name. Okay, the macrophages has different name according to their present at the different side of the body. So let's suppose if the macrophages are present inside the liver cell, inside the liver, so then we call that Kupffer cell. Okay, what we call that? We call that Kupffer cells. Kupffer cell is equal to liver. Whenever the macrophages are present inside the liver, we call that Kupffer cell. We also call that mesangial cell. We call that mesangial cell whenever they are present at the kidney. Whenever they are present at the kidney area, then we call that mesangial cells. Okay. Whenever they are present at the brain area, then we call that what? Microglial cell. Microglial cells. Okay, we call that microglial cell on that time when they are present inside the brain. Then we call that what? We call that microglial cells. Okay, and whenever these macrophages are present inside the bone, So let's suppose it is present inside the bone area. Then what we called it? We called it osteoclast. So let me write osteoclasts. We called it what? We called it osteoclast. So that's all about, that's all about the macrophages. Now let's talk about the dendritic cells. Dendritic cell, this monocyte is also converted into another cell which we call a dendritic cell. So let me draw the dendritic cell here. Dendritic cells usually look like in this form. They have their nucleus. Okay, so dendritic cell usually look like in this form, just like in the star form. If you see the nerve cell, so nerve cell is also look like in this form, but they have a tail portion as well. I'm not talking about the nerve cell, but actually it is look like in a nerve cell. So let me write, it is what we call it. We call it dendritic cell. Dendritic cells. So we also call that what? We also call it dendritic cells. Now, on what time it is converted on to dendritic cell? Again, when there is infection chances. When there is infection chances in the skin, in the mucus, in the lungs, in the nose, at that area. Whenever there is infection at the mucus of the nose, lungs, intestine, at that area, if there is an infection chances, so then this monocyte is converted into dendritic cell. Okay, this monocyte will be converted into what? Dendritic cell. Again, this dendritic cell have what? This dendritic cell have the same properties as I discussed in the macrophages. They have also MHC2, MHC1, and they have also this kind, not this CD14 and CCR3. These molecules are not present, but these things are present as I discussed. So it is nucleated, so they have also MHC1 molecule, except that they have unique molecule which we call it MHC2 molecule. Okay, so they have what? They have also these two molecules. So let me draw. So this is what? This is the MHC1, and this is what? Let's suppose at that area, let's suppose we have what? They have what? They have what? They have the MHC2. Again, this MHC2 are not present always. It may be present on that time if there is what? If the pathogen are degrading, and then this MHC2 are synthesized. Okay, they have, moreover, they have another receptors, and we also call this receptor what? We also call this receptor. So let me draw it with this red marker. They have also some other receptors. Now, this receptor is called PRRS, pattern recognition receptors. They have also other receptor which we call it PRRS, means pathogen or pattern recognition receptors. Okay, this receptor are usually involved to recognize the PIMS. Means what? They are usually responsible to recognize what? They are recognize the PIMS. Now, what does PIMS means? Stand for pathogen associated molecular patterns. What they stand for? They stand for pathogen associated molecular patterns. Okay, it is a kinds of molecules, means lipopolysaccharide. We call that lipopolysaccharides, mosines, viral nucleic acid, foreign DNA. These kinds of molecules are called what? Pathogen associated molecular patterns. So this PRRS can recognize this PIMS. So they engulf this kinds of molecules or this kinds of pathogen. Once it engulf this kinds of pathogen, then they can degrade. Let's suppose this is the pathogen which they engulf, and then they degrade it into small pieces. Now it is degraded into small pieces. After that, what actually happen? This dendritic cell will reach to the lymphoid tissue or lymphoid area. Once they reach to the lymphoid area, at there they degrade it and showcase, or they will present it or show this kinds of pathogen to MHC2. So at MHC2 molecules, there are what? there are the fragments or there are the pieces of that pathogen where they recognize it previous. Okay, after that, this whenever they presenting these kinds of pathogen pieces inside the lymphoid area or lymphoid tissue, then here the T cells are present and it is activated. T cells, let me draw here the T cell. T cells have also a receptors. So T cells, this is the T cells. T cells have also the receptors and they will recognize it. So let's suppose this hand represented the T cell and this hand, representing what? This hand representing the dendritic cell. So they have what? They have this kinds of, this is the pieces, and this T cells don't have, any kinds of the pieces, so they will recognize it. So, it is recognized. This is this upper hand is called a dendritic cell, and this lower one is what? This is the T cell. So whenever they recognize it, so then the signal is secreted from each T cells. These signals will, bring some other cells, or we can say due to this way they can activate this T cell. Activate what? This T cell will activate the innate or adaptive immunity. They will activate what? They will activate innate or adaptive immunity. So complement system, or we can say the specific antibodies are secreted from the B cells, and some other things, cytotoxic T cell are activated. All these cells or all these things are activated which are involved in the adaptive immunity, as I discussed. Okay, I already discussed the adaptive immunity. So we can say that adaptive immunity are activated. So once the adaptive immunity are activated, so then the process or under the control, or the problem is under the control. Okay. Before that, this this dendritic cell are usually involved in the non-specific immunity. This macrophage is also involved in the non-specific immunity as well. Means these two cells play important role in the non-specific immunity. Why? Because these two cells are activated against any kind of the pathogen. They are activated against any kind of the pathogens. So after that, they will actually activate the adaptive immunity. Okay. Now if we talked about their presence, so usually they are 2%. This dendritic cell inside the white blood cell, the percentage of the dendritic cell are the 2%, 0.2%. They are present 0.2% inside the white blood cell. Due to this receptor, PRRS receptor, what actually happened? They, as I told you, they recognize PAMPS. PIMPs means PIMPs, or we can say pathogen associated molecular patterns. They recognize these kinds of things which are present on the pathogen. Due to this property, this dendritic cell will distinguish between harmful cell, or we can say between the microorganisms and between the host, because inside our host these things are not present. So that's why they cannot harm our own cell. They will harm the bacteria or the microorganism which have these kinds of the property. Okay. So not only they can activate the adaptive immunity, before that they can activate what? They can activate the specific immunity. And they not only degrade the microorganism, they not only degrade or kill the pathogen, they first of all they kill the pathogen, which is which is the innate immunity portion, and after that they will showcase this or they will show this pathogen fragments to the other cells, means T cells, which is the start activation of the adaptive immuno responses. Okay. So that's all about what? That's all about the antigen presenting cell, or we can say the dendritic cell, monocytes or macrophages. Okay, I hope you understand about this video lecture. If there is something missing, or if you have any kind of questions in your mind, so you can write it into comment. Inshallah, I will give you answer as soon as possible. Okay, so like this video, share this video, and also stay tuned, because in the next video lecture we are going to talk about the granulocytes, means neutrophil, eosinophils and basophils. These three cells we are going to talk about in the next video lecture. So stay tuned. Thank you for watching.